depression are overwhelming disorders which affect millions of Americans every year. In order to better understand anxiety and depression disorders and to possibly find a cure for them, it is of the utmost importance that we are able to monitor neurotransmission of serotonin in the brain. A few weeks ago, I spoke to you about how cells in the brain communicate with one another by releasing chemicals called neurotransmitters, as shown here. If a cell releases too little of serotonin or doesn't send enough messages of using serotonin to another cell, anxiety or depression disorders can occur. Now, in order to monitor the amount of serotonin transitioning between cells, we have to use probes that are inserted into the brain. Now, a few weeks ago, I discussed some of the problems of these probes as far as them being, they have to be very, very tiny, as well as sensitive and selective to serotonin. Now, a few weeks ago, I described some of the problems associated with these electrodes, and I also mentioned that I work in a neuroscience laboratory where I tried to develop electrodes that function with certain specifications. I've been doing this for the past 2 years and focusing my honors thesis work on it, so you can trust that I understand the importance and difficulties in creating brain probes like this. In addition, the carbon fiber electrode, which I'm going to be talking to you about today, is what I most frequently use in my lab work. Carbon fiber electrodes, or CFEs, are the most effective probe to use in a mouse brain in order to have high activity and sensitivity. Now, these carbon fiber electrodes are very, very small, and they're also sensitive and selective, so they meet all of the requirements which I mentioned in my problem speech. For the next few minutes, I'm going to be going over the carbon fiber electrode and talking to you about how it works and how it's able to be sensitive, selective, and very small. Now, let's begin by discussing the anatomy of the carbon fiber electrode. JJ just passed out some examples of carbon fiber electrodes which I actually use in lab. These are the electrodes that are actually implanted into a mouse brain. Now, you can see they're very tiny, but you can see it blown up on the screen here. Essentially, carbon fiber electrodes are made out of a glass capillary, or a glass barrel, in which a conductive carbon fiber is inserted into the tip. Now, the carbon fiber on the screen here is about 7.5 microns in diameter, whereas the ones that are being passed around are 5 microns in diameter. Just as a reference, the average human hair is about 60 microns in diameter, so what we're actually inserting into the mouse brain is about 1/12 of the diameter of a human hair. Now, based on the textbook, The Mouse Brain Atlas by Franklin, I looked up some dimensions on the area of the brain which we're trying to insert these electrodes into, and it's kind of showed in gray on the screen here. And these regions have a depth of 1 to 2 millimeters. Now, intuitively, we know that microns are smaller than millimeters, so we can effectively say that carbon fiber electrodes allow these probes to penetrate into very specific tiny brain regions. Now that you understand a little bit about the size and how these CFEs meet the size requirements I discussed, let's go into some details about how the carbon fiber electrodes work. Now, according to Mark Whiteman and Kawogi in 1993, these are leading researchers in the field of electrochemistry, CFEs work through simple processes of electrochemistry or reduction-oxidation reactions. Now, once the carbon fiber electrode is inserted into the brain, as is shown here, a voltage is applied. Now, molecules of serotonin, which are very close to the electrode, actually are oxidized, which simply means they kick off or release electrons. Now, since we all know that current flow is simply the movement of electrons, the CFE measures current and sends that message to a computer. More molecules of serotonin that are present by the CFE means a higher current. Less molecules means a lower current. So we can monitor current and relate that to the amount of serotonin that's in the brain. All right, now that you know some technical details about how CFEs work, let's talk about how they can be selective and sensitive. First, selectivity. The graph you see here is actual data collected by my graduate student, Yogesh Singh, and is a graph of current versus voltage. Now, CFEs are selective to serotonin in that this graph of current versus voltage is unique to serotonin. Any of the other neurotransmitters in the brain would show slightly different curves and different peaks in different places. So to an untrained eye, it might be difficult to tell them apart, the serotonin versus norepinephrine or dopamine in the brain. But to a trained eye, you can tell the difference. Now, one part of this graph I'd like to point out to you is marked with a star here, and that is the peak current for serotonin. This current occurs at a very specific voltage that is different from the peak current location for any other neurotransmitters. The final point I want to discuss is the issue of sensitivity of these electrodes. According to a paper that was co-authored by my boss, Ann Andrews, written in 2004 in the Journal of Neuroscience Methods, extracellular levels of serotonin in the mouse brain are on the nanomolar level. This means that serotonin is present in parts per billion basis, which is very tiny. And in my own experience, as well as experience working with Yogesh, I have been able to see appreciable differences in changing currents due to these tiny changes in serotonin in the brain. So for the past few minutes, I've introduced you to the carbon fiber electrode and its uses in monitoring serotonergic neurotransmission in the mouse brain. We've talked about how it meets all the requirements of being small, sensitive, and selective. And I hope that in explaining to you how this electrode works, that you have been convinced that this electrode is an effective method of monitoring serotonin changes in the brain. Now, through the use of these tiny CFEs, neuroscientists have been able to more closely monitor serotonergic neurotransmission in the mouse brain and have therefore taken one step closer to finding cures or preventative measures in anxiety and depression disorders. Thank you.